Keith, have you had feedback from users about the new PubMLST website?
So generally, the feedback we've had has been positive. There's been a few sort of issues that are highlighted, which we'll try to address. But overall, the feedback has been very positive. So.
Okay, great. I think we're generally very happy with what Manta Ray has produced. I think it took some weeks to get there in terms of how we planned it and so on. But I think it's worth saying publicly also a thank you to Keith for the amount of effort that it took to then populate everything in the database and so on. So adding all the content wasn't a trivial matter either. So thank you for that. We have two questions. One is it possible to deploy PubMLST on my own server?
So you could deploy BigsDB on your own server. So BigsDB is the open source software. You can download all the data from PubMLST and put it into your local database, but you can't just download the databases as they exist. And we are talking about, there's a lot of data here. PubMLST compressed is about 0.5 terabyte of data. So it's not just something we could make available anyway. As a downloadable resource.
Yeah. Another question, which I think is a clarification. So it's around the analysis link. If you click on the little plus sign, it doesn't list everything. For example, BLAST. I think that's just a clarification, please.
Okay, so BLAST is from an isolate database, so that you can actually blast the genomes that are in an isolate database, not from the typing database. If you want to query the typing database, and you go to the sequence query links. Yeah. But it's not a separate plugin for that.
That's great, thank you. Mark van der Linde says, how many organisms still have their PubMLST not linked to PubMLST, but on other sites? And what do we do to integrate them?
So if they're hosted on another site, like, say, Pasteur, which also uses BigsDB, then we have been in discussions as to how we could actually mirror those, so that we can actually have the same data available on both sites, so that it's all accessible from a single resource. If schemes are not, if they don't exist within a database, then there's not a lot we can do. I mean, we are open to people asking us to host their schemes, and we will do that if they do that. And we're constantly adding new schemes in there, but people have to come to us to host them. Yeah, that's great. There's a question by Frederica also about anything that has changed for curators of the database. So generally, we're trying to improve the curation experience, to try and all the things that can be done sort of automatically. We're trying to do that. So a lot of that is trying to improve sort of metadata completion and validation, so that sometimes you can't put a record in where you've got sort of contradictory information in the field. That work is still undergoing. It's still underway. The main part of the BigsDB that has changed is really only the interface. The actual functionality hasn't changed with this redesign, but we are constantly developing new functionality to improve curation.
Yeah, that's great. Richard asks, is the code for the front end also open source, and are you continuing development of it in-house?
So the front end, all the bits outside of BigsDB, is actually Drupal, so that is open source, but it's not our code. Obviously, the artwork and stuff is all proprietary to PubMLST, but the actual code, yes, it's Drupal, standard content management system.
Yeah. Ahmed says, at the beginning of the lecture, only Campylobacter coli and Nonjejuni were contained in the list of organisms on the new PubMLST. Can it be used for other species of Campylobacter? So
there is a Nonjejuni coli database as well. So if you search for Campylobacter, then you'll see that page will come up. So these are separate schemes. We have to differentiate really between Nonjejuni coli and all the other sort of commensal campys and environmental Campylobacters. But yes, they are on the site.
Yeah, that's great. Rafael Ibrahim says, what format of sequence is PubMLST accepting? FASTA files or FASTQ files or both?
Yeah, so FASTA. We only deal with assembled data. We don't handle short reads on the site.
Yeah. Okay, so Mustafa says the analysis units to analyze whole genome sequence data, for example, core genome MST data, seems to be quite low. Any plan to expand that? I think maybe options for analyses, I think.
Well, it depends what you're trying to do. I mean, you can certainly analyze all the isolate data based on whole genome data. So you can draw minimum spanning trees. You can do forensic analysis of allelic differences across whole genome, across any sort of whole genome scheme within the isolate database. So I think you have to sort of make a distinction between the typing side and the sort of isolate repositories. But certainly within the isolate databases, there are a lot of tools for analysis of whole genome sequences.
Yeah, I think maybe some kind of poking around to see what things are there might be useful for users. There is actually quite a lot that you can do within the website. Hello, Joao. Joao says, nice website redesigning. Concerning functionality, can we use the REST API to submit new alleles to a given schema?
Yes, you submit them to the curator queue. So then a curator will either assign them, or if they're sort of easy to assign, like a core genome MLST allele is usually fairly easy to assign if it's if it's got start and stop codon in the right place and everything, then that can be assigned automatically. But yeah, that's all automated, and we have links now from Entrabase of automatically submitting new data directly into PubMLST through the API, and a few others have begun to do that.
So G1 says, can PubMLST perform core genome MLST and whole genome MLST analyses for all the supported species?
It can do it for all species for which a scheme exists.
Correct.
We only host what people have actually developed.
Okay, there's another question around the raw reads. So this person loves the new web for the UI for PubMLST. Continuing on the raw read question, are there any plans to improve linkage with the short read archive or ENA and other ENA hosted databases like biosamples? Different ways to encourage users to submit raw reads to those databases and link the reads to the entries on PubMLST. That's a loaded question.
So many of the databases will have fields for accession numbers that link out to the ENA. So you can you can go to a record and then you can download the short read data directly from the ENA. And of course, that's all available through the API. So you will be able to sort of link that. You'll be able to automate that if you want. And likewise, biosamples are also, in many of the databases, we have fields for the biosamples. All the databases, we can completely customize which fields we include. So it tends to be the databases that have a lot of genomic data that tend to have more fields that link out to accessions in other databases. But we can certainly add any field that we would need to. So yes, we are sort of improving the links out to other data resources as much as possible.
Okay, I'm going to take one more question and then we'll have to move on to the next one. But it's a question maybe, Keith, that you could sort of generally answer. So what problems can one face if they're working with draft genomes instead of complete genomes? Is there anything you want to say about that, really?
I mean, it depends what you're trying to do. I mean, generally, if you're trying to extract your typing information, then that works absolutely fine with draft genomes. Most of the genomes on the database are draft genomes. They don't exist within a single contig. They'll be in, maybe in a few hundred contigs, depending on the species. But generally, most of the analysis can be performed on those draft genomes just as well you can on a sort of complete, complete genome. Obviously, you will have issues about identifying synteny between loci. If they're on different contigs, you can't tell that. But as long as the genes are actually fully within a contig, then it doesn't really hold any problems for analysis.
Okay, thank you very much, Keith. There are some more questions in the chat, but I think in the interest of time, we'll move on. But maybe, Keith, you could have a look and even answer those directly if possible.